So this is chapter 6, which is, I just Googled eating, and this was the picture that came up. One of the first ones. I thought it was really disgusting, but it applies. Okay, so we're going to be talking about how cells take food sources, a fuel source like sugar, like I think this is a chicken burger, and release the energy from it and make ATP. We all know that ATP is what living cells require. So all cells require energy, and they require energy in the form of ATP, which we've talked about in previous chapters, in order to drive forward endergonic reactions, so things like active transport, things like protein synthesis, things like protein transport, things like muscle contraction, all kinds of things that require energy to go forward are going to require ATP. All cells are going to use energy from food to make ATP. So ATP is produced in cells by breaking down organic compounds in a process called cellular respiration. So you've probably heard the word respiration before, and you probably think about breathing. Sure, that's one type of respiration. Cellular respiration is the process of breaking down a fuel source to generate ATP. Animals get these fuel source compounds from other animals. Some get them by eating plants. So here's one eating plant source. Here's an animal eating an animal source of fuel. And plants will get these organic compounds through photosynthesis, which is what we talked about the other day. So just to kind of make sure we're on the same page, do plants carry out respiration, cellular respiration? Okay, that should be enough time. Looks like most of you have answered. Okay, so good, most of you got it right, but a few of you picked some of the wrong ones. So let's just talk about some of these. So no, photosynthesis has the same function in plants as respiration. Is that true? No. What does photosynthesis do? Photosynthesis produces what? Okay, but that's not the reason for photosynthesis, though. Yes, it's one of the byproducts. But what's the whole goal of photosynthesis? To produce what compound? Sugar. To produce a sugar source. Okay, so photosynthesis produces sugar using energy from sunlight, and oxygen is a waste product of it. What does respiration do? So what does it create? It uses sugar as its fuel source, but what is it creating ultimately? Which was on the last slide? ATP. Okay. There's only one slide to go, but I know we're warming up. We're getting into the day. No, it's okay. Do you think plants just don't need energy? Actually, no one answered that, so that's good. That's wrong. Plants need energy. Plants have endergonic reactions. They need to produce proteins. They require cellular respiration as a way to get rid of extra CO2. Do you think that could possibly be true? Not really. Yes, it consumes, well, actually, no, because it doesn't get rid of CO2. It actually produces CO2, cellular respiration. And, really, this is the only real reasonable answer. Yes, they require ATP, just like any other living things. And respiration is a process that generates ATP. So plants are living, so they need to do cellular respiration. So any living thing does cellular respiration. Doesn't mean that all living things have mitochondria, which we'll talk about what that is as we go through this chapter. There are other ways of doing cellular respiration that don't require oxygen, that don't require mitochondria. But all organisms need to convert a stored fuel source or a food source into a usable energy source like ATP. So they need the process of cellular respiration. So this is a huge common misconception. I hear it all the time from students. Oh, well, plants do photosynthesis and animals do cellular respiration. Well, yes, you're correct in that, but plants also do cellular respiration. So Everything does cellular respiration. Photoautotrophs do photosynthesis as well. So 70% of you got that right, but just be careful with that. Don't forget that plants also do this process. So we'll often probably approach this from the animal perspective because we're animals and we're very human-centric, but all organisms are doing this as well. Okay, so cellular respiration is the process of using the energy that's stored up in food to make ATP. And as I said, there's several mechanisms for doing this. We're going to talk about a different kind of mechanism near the end of this chapter, probably in the third lecture on this chapter. We're going to spend most of the chapter talking about one mechanism for releasing energy from food to generate ATP, and that's a process called aerobic respiration. The word aerobic respiration means with oxygen. So aerobic is with oxygen. We're going to be talking about anaerobic later on. What do you think that is? That's without oxygen, yeah. So we're going to talk about an is without, so without oxygen. So aerobic is with oxygen. And the equation for aerobic respiration is the opposite equation as photosynthesis. It's glucose plus oxygen yields carbon dioxide, water, plus energy in the form of ATP. So we're going to see that oxygen in humans, we get that by inhaling the oxygen. Oxygen is then transported to all of our cells by our bloodstream. The whole reason for having a circulatory system is to get that oxygen to all of our cells. It also transports the sugars as well, but it transports the oxygen to all our cells. Inside of our cells, we're going to talk about something called a mitochondria, which is vital for performing a lot of the steps of this reaction. And that's going to convert that glucose using oxygen into carbon dioxide and water. Carbon dioxide is then going to go back into our bloodstream and be exhaled. So you are inhaling the input as well as you're eating the other input of cellular respiration, and you are exhaling the output. And you could think you're also exhaling moisture too, and that's the two outputs. So you eat this, you breathe in this, and you breathe out those products. And this is going to produce ATP, and ATP is going to drive cellular processes. So energy in food is going to make ATP. And this happens in autotrophs, this happens in heterotrophs. Autotrophs just happen to make their own food, whereas heterotrophs have to eat it. They have to consume it. Okay, so like photosynthesis, respiration is a big redox reaction. It's an oxidation-reduction reaction. You are transferring electrons from glucose to oxygen. So if I write that equation up on the board, we have C6H12O6 plus 6O2 gives us 6CO2 plus 6O. Where am I going? Here we go. H2O plus ATP. And you can see that electrons have been transferred once again in the form of transferring of hydrogen. So notice where the hydrogen is in both of these. And the hydrogen is moved over there to make H2O. So what's happening is the hydrogen, the electrons and the hydrogens are being stripped off of glucose and combined with oxygen. This is an extremely exergonic reaction, which means it releases a lot of energy. It's, I'll show the number later on. It's like 683 kilocalories or something per mole. It's a huge amount of energy that's released in this process. If it was released all at once, it would destroy the cell. This is actually a very similar overall type of reaction as what rocket fuel does. So you take liquid you take hydrogen and oxygen, you mix it together with a spark, and you get an explosion, explosive release of energy that propels rockets into outer space. So it's enough energy to do that. But your cells are not destroyed when you eat, right? That would be really bad if every time you ate a hamburger you exploded. So in order to prevent this explosive kind of damaging release of energy, your cells will release the energy in a series of small reactions, small exergonic reactions, each one which is going to strip off a little bit of energy. So slowly, the energy from those electrons is going to be lowered as ATP is generated until you can finally make water at the end of the process. So kind of an analogy to this would be if you were sitting at the bottom of a giant waterfall, like Niagara Falls, you would get crushed. But if you gradually let water go down a series of smaller waterfalls, you can go the same distance and you can stand at the bottom and you're not going to get crushed anymore. So each of those little reactions is not going to be explosive release of energy and wasteful release of energy. So it allows your cells to capture a large percentage of the energy stored in food through what's called a stepwise or a controlled release of energy during cellular respiration. So, which means we're going to have to learn about several reactions. All right. So, just again like photosynthesis, there are several steps to cellular respiration, and we're going to start talking about them today and get through maybe the first, maybe the second one. We'll see how far we get through today. So respiration occurs in 3 steps: glycolysis, the Krebs cycle, and electron transport. There's actually a short little step between glycolysis and the Krebs cycle we'll talk about as well, but I won't really give it a name. It'll just be the transition step, or let's say the acetyl-CoA step. In the first 2 steps, glycolysis and the Krebs cycle, energy is going to be stripped off of your fuel source. So glucose is going to be our fuel source that we're going to use for this. And it's going to be transferred to electron carriers called NADH primarily and FADH2. So if you remember NADPH, these are very similar to that. So even their name's pretty similar. So NADPH was in photosynthesis, so P, photosynthesis. NADH, FADH2 are in cellular respiration. They are electron carriers. They are intermediate energy storage molecules that are going to be carrying those electrons and that energy from one reaction to a later reaction. The energy that was picked up by NADH and FADH2 is then going to be converted into ATP in the final stage, the electron transport stage. So we're going to see that glycolysis occurs outside of the mitochondria. The products of glycolysis, which we'll see as pyruvate, is going to go through a transition step and then enter the Krebs cycle, which occurs inside the mitochondria. All of those stages are going to be producing these energy carriers, mostly NADH, but a little bit of FADH2 as well, which serves a similar role. Those energy carriers then take that energy to an electron transport chain. It's going to be very similar to the electron transport chain we saw in photosynthesis. That electron transport chain is going to produce a ton of ATP. And here, ATP is the ultimate goal, so that's when we're done. So we're going to be looking at each of these steps in a little more detail as we go through this stage. So glycolysis, Krebs cycle, and electron transport. And I will expect you to know pretty much the inputs and the outputs from each of those cycles. So what goes in, what goes out, what's produced, what's required. And that's really the ultimate goal. I won't require you to know all the little intermediate, minute, detailed chemicals and different enzymes and things like that. It's just, that'd be a little ridiculous. Okay, so the mitochondria is vitally important for this process, these steps to go through, so we need to know a little bit more about the shape of the mitochondria. So just like the chloroplast structure was really important for photosynthesis, mitochondria structure is, and its shape allows it to perform the reactions of respiration really efficiently. Mitochondria consist of two membranes. They have an outer membrane and an inner membrane, and the outer membrane is basically just a sphere. It covers the whole outside of the mitochondria. The inner membrane is highly folded. So it's got all these different folds in them called cristae, and they create this huge surface area. And we're going to be seeing that the last step of respiration, electron transport, is going to occur on this inner membrane. And by having that huge surface area, it allows your mitochondria to be packed with a lot of electron transport chains. So we're going to be looking again, just like we did in the chloroplast, at just one, but there's hundreds of thousands of them throughout mitochondria, and there's several mitochondria in your cells. The space between the two membranes we call the inner membrane compartment, or the inner membrane space, and we're going to be talking about what happens in this space here. It's going to be, again, very similar to what happens inside the thylakoid. And then the inside, so what's inside the mitochondria, is called the matrix. So if I was to draw it here, and draw a little fewer folds so you can see it, this would be the matrix, and this would be E there. So the inner membrane compartment is between the two layers. The matrix is in the very center. So just give us some geography. So when we talk about what happens where, we're going to see that glycolysis, the first step, occurs out here. The Krebs cycle occurs in the matrix. And then electron transport will occur on this inner membrane. Okay. So, first step of cellular respiration is glycolysis. Glycolysis is going to be the first reaction that breaks down glucose. It's going to break glucose into another sugar called pyruvate. So glucose is a 6-carbon sugar. Pyruvate is a 3-carbon sugar. So when you split glucose into pyruvate, how many pyruvates do you think you make? Yeah, two pyruvates from one glucose. So the first stage of respiration is the splitting of glucose, breaks glucose into 2 molecules of pyruvate. It occurs in the cytoplasm, so outside the mitochondria, and it has 2 phases to it. It has an energy investment phase and an energy harvest phase. So in the energy investment phase, your cell is actually going to burn a little bit of ATP. And the energy harvest stage, it's going to be making some ATP, as well as making some other energetic products as well. So if you remember back to our chapter on energy, when you have an exergonic reaction, like the breakdown of glucose, what often has to happen before a reaction can get going? So does the reaction go like that? It usually often goes up first, or it often requires activation energy. So this energy investment phase, that's what it's doing. It's investing some energy to get the reaction going, and then the energy harvest phase is going to make back that energy, basically. So you give up a little bit of energy to make some energy. Okay, so I took this from your book, this next slide, which is a picture of the steps of glycolysis. I don't know why they did it this way. It's very overwhelming, right? So they broke it down into 10 easy steps of phosphate transferring from ATP to glucose, some rearrangement, second phosphate, 6-carbon intermediate, creating 2 3-carbon intermediates, oxidation, substrate level phosphorylation, rearrangement, removal of water, and then another substrate level phosphorylation. So if you're really keen on this, go ahead and learn all these steps. They're a lot of fun. Each single one requires its own enzyme to catalyze it. There's a lot of names you can learn. You can probably even look up the chemical structures of these if you really want to do it. But really, there's a lot less of this than I want you to know. Mostly I want you to know what goes in and what comes out. And that requires you knowing the name of the first thing, glucose, the name of the last thing, pyruvate, and then all the energetic products that are made throughout the reaction, which include ATP being invested here. So you can see ATP is on the back end of those arrows and ADP is on the front end. So how many ATP are invested in that first, in an energy investment step? 2, yeah. So we invest 2 ATP. And then on this side, this is where we get stuff back. This is the energy harvest stage. So how many ATP are coming back to us? 4. So we invest 2, we make 4. So how many ATP does glycolysis produce in the end? 2. So we burn 2 to get 4, so we're up 2 in the end. It also produces 2 molecules of NADH, so 2 electron carriers. So here I've squished this down a little bit further, and this is really all I want you to know about glycolysis. Not all those intermediates, not all the names of everything, not all those 10 different steps, but basically this. So energy investment phase invests 2 molecules of ATP to get the reaction started. So that's going to overcome that first hurdle. Energy harvest stage now makes 4 ATP and 2 NADHs in the process of creating 2 pyruvates. So we could draw this. The way I usually draw it is this. So glucose, which is C6H12O6, is converted into 2 pyruvate. And you have a net 2 ADP and 2 NAD plus that are being converted into a net 2 ATP and 2 NADH. Okay? That's really, you could boil it down to that, is that this is what goes in, this is what comes out. This is what goes in, this is what comes out. Okay? And that's glycolysis. So why did I write net here? You know what the word net means? Yeah, so it's all together. So all together I've if I counted up total, I've burned 4 ADP to get 4 ATP here, and I've used up 2 ATP to get 2 ADP. But if you add that all up and you sum it up, you are making 2 ATP, because you burn 2 to get 4, and you're making 2 NADHs. So these are the 2 energetic products that will be used later on in the reaction that have been made through glycolysis. Okay? So that's also on this right side of your graphic. The net input, the net output. Okay, does that make sense? Questions on glycolysis? Yeah.
What does it say about the 2 NADH?
2 NAD plus. So NAD plus. It's like NADP, or sorry, NADP plus. It's the same concept. It's without the electrons and without a hydrogen. And then this is it carrying the electrons. So this is also energetic. So I could put a little star on this if you want. There you go. They're both energetic products. Okay. All right. So here's another question for you. Okay, the oxidation, so stripping of electrons from glucose to carbon dioxide and water, releases 683 kilocalories per mole of free energy. So it has a delta G of negative 683 kilocalories per mole. The reduction of NAD plus to NADH requires 53 kilocalories per mole of free energy. Why, then, have we only produced 2 molecules of NADH during the glycolysis process when it appears that many, many more can be produced? So why do you think that is? Well, it's fine. Most of you are wrong, but it's fine. That's the way it works. So I use these to illustrate a point, so it's fine. Okay, so here's what you guys said. So most of you said C. Some of you said ATP, and some of you said pyruvate, and very few of you said it was just very inefficient. So let's take care of A first. So we didn't talk about this, but you're right, some ATP is produced. So in that reaction, we've got glucose being converted to pyruvate, ADP to ATP, NAD to NADH. So we're making 2 NADHs, so we'd have times 2 of this, which would be 106 kilocalories per mole. So we're left with 577 kilocalories per mole left. So ATP, ADP to ATP is about 7 kilocalories per mole. It's actually much less energetic than NADH. So NADH is a much higher free energy compound than ATP is. Even though ATP is so important, it's only a small amount of energy. So we had 2 of those, so that's another 14. So we've successfully, so that's 14 kilocalories per mole. So we're left with negative, what do we have? So that's 120, so 563. So somewhere there's 563 kilocalories per mole of energy somewhere, right? So it's not in ATP because ATP is only about 14. So it's not enough. So NADH, ATP, that only accounts for about 120 kilocalories. There's still 563 if I did that math right. So is it that glycolysis is very inefficient? Well, it does lose some of the energy as heat. Every time there's a reaction, like we've learned before, there's a little bit of energy lost as heat, but not that much. That's a huge amount of energy lost as heat. Every time you ate, you'd be burning up. So you do get a little bit warmer, right, when you eat usually, but you don't catch on fire. So it's not that. So is it C? Is it the glycolysis consists of many enzymatic reactions, each of which extracts some energy from the glucose molecules? And that is true. It does consist of many different reactions, and they all extract some of it, but they're extracting it into ATP and NADH. If they're not, then that energy is being lost as heat. That energy goes somewhere, because remember, energy is not created, it's not destroyed. So what we're left with, if it's not here and it's not here, then it's here. So we have 2 molecules of pyruvate. Those are 3 carbon sugars. They still have a ton of energy left in them. So the answer to this question, which a quarter of you got, is that most of the free energy available from the oxidation of glucose is still present in an energetic compound that has yet to be broken down called pyruvate. It's one of the products of glycolysis, and we still have the Krebs cycle and the transition state before the Krebs cycle to release that energy. So there's still a lot of energy left. And the reason why I kind of highlight this now is just to show that we've only stripped off a little bit of energy, and we actually, if you look at the energy that we've stripped off that is now in a usable form that our cells could directly use to drive active transport, to drive muscle contraction, things like that, we've only produced 14 kilocalories out of 683. So if we stopped here, this would be a highly inefficient process. We'd be taking a hugely energetic compound and only using a teeny bit of it, right? You'll see there are organisms that do this. That's all they do, and that's where they stop. And we'll get to that near the end of the chapter. But right now, I want to talk a little bit about the next step. And that's probably all we'll do today. Okay, so there is a little transition step that occurs between glycolysis and the Krebs cycle. And so I'll quickly talk about that with this slide. As pyruvate moves into the mitochondrial matrix, so glycolysis occurred outside the mitochondria, the Krebs cycle is going to occur inside the mitochondria. As pyruvate moves in, it's going to be broken down a little bit into a product called acetyl-CoA. CO2 is lost, NAD plus is converted to NADH, and the remaining carbons combine with something called coenzyme A to make acetyl-CoA. It's basically preparing this molecule to enter the Krebs cycle. So we've created a little more NADH. And remember, we had 2 pyruvates, so if each one produced 1 more NADH, we've now produced 2 NADHs. So if pyruvate was a 3-carbon molecule and I lost 1 carbon dioxide per pyruvate, I now have a 2-carbon molecule, acetyl-CoA. So this is a very short little step. We sometimes call this a transition step, or we call it pyruvate oxidation is another word you might hear for this, because you're stripping off a few more electrons from pyruvate and prepping it for the Krebs cycle. Okay, so we now have acetyl-CoA. Still has a lot of energy, so we've stripped off another 106 kilocalories from that, but we still have 400 and some odd kilocalories left in these 2 acetyl-CoAs. So what the Krebs cycle is going to do is it's going to strip off the rest of that energy. So we're going to take the remaining energy from acetyl-CoA and convert it into more energy carriers and more ATP in a cycle called the Krebs cycle. The Krebs cycle should remind you a little bit of the Calvin or the carbon cycle from photosynthesis. It's pretty much the same thing in reverse in many senses. You're going to be putting a sugar in and getting energetic products out, whereas remember for the Calvin cycle, you put in energy, ATP and NADPH, and you got sugar out. So this one, you're going to be entering acetyl-CoA and getting out NADH, FADH2, and ATP. So when acetyl-CoA enters the Krebs cycle, the first thing that happens to it is it binds with a sugar molecule that's already in, producing a molecule called citrate, which is a 6-carbon molecule. So just like what happened in the Calvin cycle, something enters it and combines with something that's already there. So you'll often also see Krebs cycle described as the citric acid cycle, because citrate, or citric acid, is the first thing that's made when acetyl-CoA enters the cycle. So I have a picture of this on the next slide that should help describe this. Okay, so citrate then will go through a series of reactions that rearranges, that oxidizes it, and every time it gets broken down, CO2 is released, and it produces ATP and the electron carriers NADH and FADH2, as well as it produces a bunch of hydrogen ions. And eventually, the process will regenerate the original 4-carbon molecule that first bound with acetyl-CoA, allowing the cycle to continue again. So just like we saw in the Calvin cycle, you're going to ultimately regenerate the starting product, but in the process, every time the cycle goes through, it is going to be producing ATP, NADH, and FADH2, which are the energetic products of the Krebs cycle. Okay. And the next slide, we'll talk about numbers in terms of these things. Okay. So here it is in a picture form. So acetyl-CoA is coming in on the top there. It's mixing with a four-carbon sugar that's already in there. It's called oxaloacetate, but don't worry about that name. To form citrate, that's the first step. It combines to make a large 6-carbon sugar. This 6-carbon sugar citrate is going to be broken down. So every time it loses a carbon, every time it's broken down a little bit, carbon dioxide is released and NADH is formed. Then carbon dioxide is released and NADH is formed again. And then eventually ATP is formed and FADH2 and another NADH. So these are all produced as the energy is slowly stripped off of citrate, eventually reforming oxaloacetate again to allow the process to continue. It can then combine with another acetyl-CoA. So the input into the the Krebs cycle is 2 acetyl-CoAs, basically, as well as a lot of lower energy forms of your energetic products. And the output are 2 ATPs, 6 NADHs, and 2 FADH2s. So there was a lot of energy still stored up in those 2 acetyl-CoAs. It's now converted into a lot of NADHs and FADH2s, which are energetic carriers. And remember, each of those NADHs represents about 53 kilocalories. So 6 of them, we have about 300 kilocalories. And FADH2s are a little bit less energetic, but they're close to that. And 2 ATPs, that almost represents all the energy that was stored in those 2 acetyl-CoAs. So we've now completely broken down the glucose that we started with from a 6-carbon sugar down into single-carbon carbon dioxides, and the energy that was in those glucose bonds is now converted into either ATP, but only a little bit. We have about 4 total ATPs now, but mostly into NADH and FADH2, into electron carriers. Okay. Okay. Questions on the Krebs cycle? Just to remind you a bit of the Calvin cycle. Pretty similar. The Calvin cycle was kind of the opposite input. You input carbon dioxide, whereas here you get carbon dioxide out. You input ATP and NADPH, whereas here you get ATP, NADH, and FADH out. And in that one, in the photosynthesis, you got sugar out, whereas here you put sugar in. Right? So it's very much, if you just look at the overall picture and not at the individual molecules, it's the opposite process. Okay, so be very careful with these 2 chapters, especially since you're going to be studying for them both for the same test. There is a lot of overlap between them. Make sure you can separate the 2 in your mind and know what goes where. So you use the terms NADPH only with photosynthesis and NADH with respiration. Right. Okay. Okay, so just to kind of summarize, let's say, so we started with 1 molecule of glucose, let's say. How many molecules of NADH are now available for the electron transport chain? So let's see if you can look back through your notes and count up everything that happens in each step. Okay, final answers? All right. Well, 7% of you were right. So let's see what you missed. Okay, so the first step of cellular respiration is glycolysis. How many NADHs are produced in glycolysis? 2, right? So we had 2 NAD pluses to 2 NADHs. So we have 2, right? Okay, what happens next? Where's the next place we see NADH produced? Okay, in what step?
Intermittent step.
Yeah, so this transition step, how many are produced there? How many NADHs per molecule of glucose? 2 total, because we went from, we have 2 pyruvates, and each of those is going to produce 1. So we have 2 more there. Okay, and then the Krebs cycle, how many are produced there?
6.
So I got 2, 4, and 6. 6. There's 6 there. So this equals 10. All right. So here, I'll unveil this. So you guys were not very close, but you counted them up, but you might have missed 2 somewhere. It's not ultimately, the number's not that important. I just want you to know a little bit what happens at each step, and so you know that each of them happens. So each round of those the Krebs cycle produces 6 NADHs, produces 2 FADH2s. But if you look at just the NADHs, you think NADH, each NADH is worth 53 kilocalories, and we have 10 of them. That's 530 kilocalories out of the 683. So the bulk of the energy that was in glucose is now in NADH. There's a little bit in FADH2, which is another electron carrier, and we've only gotten about 28 of those kilocalories in ATP. So we had 2 here and 2 there, so we have 4 total. So we still have a lot to do. But just to summarize, here we go. Go to the next slide. Here we go. So 1 molecule of glucose produced in glycolysis 2 ATP and 2 NADHs. The acetyl-CoA formations, that transition state, produced 2 NADHs. And the Krebs cycle produced 2 ATP, 6 NADHs, and 2 FADH2s. So again, the bulk of our energy is in electron carriers, mostly in NADH, a little bit in FADH2. And we've only generated a little bit of usable energy, the ATPs. So the next step, what we're going to talk about next week, is what happens to these electron carriers. How do we take the energy from these electron carriers and use it to generate the bulk of ATP? Because most of the ATP produced in cellular respiration is done in the last step, the electron transport step. So this is where we'll stop for now, and we'll continue on Monday with seeing what happens to these molecules. Okay? That's it.